Lishawa, a part of Team Typha, the person who leads all the work. I'm nominally a co-PI, but I really, Shane does everything. Nancy Tuckman is the lead PI along with Beth.
Depending on the grant, but yes, definitely. We have a broad group. I'm kind of a yeller too, so if you squint as I talk, whoa. I'm sure y'all try not to blast your ears out, but it's the last talk of the morning here, so maybe I should keep you awake. Okay.
Here's your slide advance. Put them down. There you go.
So as Newt mentioned, I'm part of this group that has been termed Team Typha, and Typha emerged from the womb in roughly 2003, and I've been involved working full-time on this project, on this work since about 2008. So we have a very broad group, and it's getting bigger and bigger by the week, it seems. And I think that Mark sort of had a nice segue into our work in that I think our work kind of stands out or is a little bit different than a lot of the research at the biostation because we're really focused on the ground action and on actual ecosystem management, and so it's sort of a little bit of a different angle, and I'll take it from there. So I have a different title in the program, and this I after thinking about what I was going to present today, instead of just doing a data talk, I decided to do more of an umbrella overlook of what our group is working on and some of our current successes, and then moving forward some of our projects that we're just starting to begin. And adaptive restoration, I think, is a good kind of umbrella term for what we're working on and what's kind of guiding our science. Okay, I'll just go through this. So this is a very simple diagram illustrating what this, this seemingly jargonist term is, but adaptive restoration essentially is a way for science to dictate how management happens on the ground using hypothesis-driven, question-driven restoration practices. So essentially, we are looking at problems, ecological problems, and designing experiments to, based on hypotheses on data, to deal with those environmental problems, and implementing, various scales of restoration on the ground. It can be very small scale, and then scaling up to, we're working in the scale of hundreds of acres at this point of actual restoration practice. So it's really a nice vehicle for creating large-scale ecosystem, studies where you can do large-scale manipulation through this adaptive restoration process. So I'll return to this figure, but and I also want to say that sort of building on something that Newt kind of described in a little press piece that we had this summer is that this, I think our project represents the outward-looking nature of the biostation, and I think that we have this broad network of regional partners now, and a lot of the scientists that are involved are from the station, but we're plugging into all of these local groups in the UP and in northern lower Michigan, and we're really penetrating, invading the region through this work. Just real briefly, to identify the principal problem that's kind of driving the work that we're working on. So Great Lakes wetland ecosystems, this is a figure that Denny Albert put together, who is, along with Nancy Tuckman and Dennis Albert's at Oregon State. He's a longtime biostation faculty member, and then Nancy Tuckman also is a longtime biostation faculty member. And the three of us sort of kind of developed this more management style research at the beginning in like 2010. So anyway, this is a slide from Denny Albert, and what we're looking at here is the Lake Michigan shoreline, or just in the state of Michigan, the amount of shoreline, and he just spread out a line demonstrating how much shoreline there is relative to the east coast of the United States. So a lot of times, if you're not in the Great Lakes region, people don't appreciate the overall extent of the coastline. It's really a huge area. And to focus in on wetlands, our work is in Great Lakes coastal wetlands. there are approximately 212,000 hectares of Great Lakes coastal marsh and wetland, Great Lakes wide. The majority of those wetlands occur in Lake Huron and Michigan, and then the other Great Lakes have a slightly less overall area of coastal marsh. And these ecosystems are hugely important for, they're more important than their size alone would sort of describe. So that seems like a huge number, but if you look at overall wetland area in the eastern United States, that's just a small fraction of the overall wetlands. There's a huge amount of inland wetland that would dwarf that number. But the habitat values and the ecosystem service values of these ecosystems make them more important than just their area alone would sort of describe. There's many different plant species, animal species, threatened and endangered species, and then the ecosystem services are really critically important. A large percentage of the fish in the Great Lakes reproduce or have some part of their life cycle in these coastal wetlands, so it's really important for the whole Great Lakes ecosystem. So we have been focusing on invasive plants in the Great Lakes, and Nancy Tuckman's work, starting back with Team Typha's birth, was looking at invasive cattail, focusing on Cheboygan Marsh, which is one of the closest Great Lakes coastal wetlands to the biological station. And the species that's dominant there is Typha glauca, which is a hybrid cattail. But the genetics are kind of messy, and so we have, there's some back crossing with this other invasive species, Typha angustifolia, and so we group these together as invasive Typha. It's just a lot, I guess, more accurate than to describe them as individual species. Another species that's of great concern, and I'm sure you've heard a lot about it, is Phragmites australis, which is becoming more and more common, and over the last decade has really expanded a lot in the Great Lakes. And then the third species is reed canary grass, Phalaris rhodonacea. And these are the three biggest, most expansive wetland emergent invasive plants in the Great Lakes basin. Okay, so just to give you a sense of the numbers of coverage, so Dennis Elbert he's part of the Great Lakes Coastal Wetland Monitoring Program, which has been a huge effort by EPA to evaluate a whole range of biota across the Great Lakes. And using his data Great Lakes wide, I'm really excited about this. So, but we just are able to finally draw some conclusions Great Lakes wide with field-based data on coverage of these different plant species throughout the Great Lakes. And to put this into perspective, so invasive Typha has about 30,000 hectares of, this is dominant cover. It's not just presence, this is where the species is actually dominant. And Phragmites has about the same amount of cover, Great Lakes wide, and then reed canary grass has about 20,000 acres of coverage. So we're talking 80,000 hectares of invasive plant dominated wetlands in the Great Lakes region out of 212,000 total. So it's a major environmental issue here. To break that out by individual lake basin here, so this is broken into two different primary vegetation zones. So in the emergent vegetation zone, so that is where you generally have standing water dominated by bulrushes historically, and this is broken out by different lakes. So for instance, in Lake Erie, approximately 70% of the emergent wetland is dominated by one of those three species presently, and in the meadow, about 50% of what's extant is dominated by invasive species. Lake Ontario is even worse as far as the amount of area dominated by invasive plants, and the other three lakes come in at some lower values. But this is just kind of a shocking figure to me to see that. We're lucky at the biostation to be able to be working up in northern Lake Huron, where these invasive plants have not come to totally dominate every last inch of the coastal zone. Okay, so there's our problem. We have an invasive plant problem. And this provides a really important spot to launch on-the-ground research. So what I'm going to highlight now is a couple of recent accomplishments, a couple of recent papers that came out without going into great detail. that illustrates some of our recent work on adaptive management research in these cattail areas. So one of the things that strikes us and has struck a lot of different people when you look at these invasive plant populations is they're hugely abundant, they're giant plants, they're really productive. So we wanted to look at the possibility of using harvesting with pairing harvesting these plants with utilization of the plants for bioenergy as a possible solution for trying to deal with these plants at this huge scale. And so in beginning in this is 2012 or 11, we did a experimental harvesting experiment out of Cheboygan Marsh, where we hand removed cattails and we gathered litter and we hand removed rhizomes from a certain subset of plots, and then we monitored plant response following that work. And this image here is 1 year following harvesting. So you can see in the background, this is a dense cattail stand with litter that was standing dead litter over 1 meter tall and very low plant diversity. And 1 year following harvesting, we got this just robust seed bank response in some of our plots. So in this plot here, we've got native sedges and rushes and annuals, a wide variety of plant species. And as I was sampling this quote that I had on an old SNRE t-shirt from the 90s was running through my head as I was sampling. And so something that's just kind of always kind of amazing to think that there's this whole community of organisms just waiting for the environmental conditions to be right for them to germinate and come back and recolonize these areas. Okay, just moving on. So here's some data to back up that image. Plant diversity increased following harvesting. These are several different measures of plant diversity over 2 years. We had basically a 2-year increase from year 1 to year 2. We got greater plant species richness and diversity following harvesting. So that's really encouraging. So at that point, we had data to support this as a management tool. We went back and we said, okay, let's try to do this at a bigger scale and see if we still see the same patterns and see if we can ask some more interesting questions. Okay, a second problem that we've identified, it's very really stark, is that 94% of land managers use herbicides to control invasive Phragmites. So it's almost the only thing that people do to deal with Phragmites and cattail. What are the implications of that? So that drove some other questions that we began looking at. And we found, and this is from Lawrence 2015 paper, Beth Lawrence was a postdoc in Tubman Lab, and she still is at the biostation and collaborating on all this work. We found that following herbicide application, we had an increase in a whole range of nutrients up to a year following when we applied these herbicides. So we had increase in nitrate and ammonium, and total nitrogen, phosphorus, and potassium were all significantly greater a whole year following herbicide application. And so what's the problem with that? And obviously there's a lot of problems with that, but the nutrients themselves are driving a lot of the increase of invasive plant cover in the Great Lakes and elsewhere. So you may be dealing with the problem of your invasive plant momentarily from a plant community perspective, but it's actually creating conditions that are favorable for reinvasion by other species. So that's just not a very holistic way of dealing with management. I think that we think that harvesting is maybe a more appropriate technique than herbiciding for dealing with these plants. Okay, so current research. In 2015, you can see the regional scope of our work. We have a bunch of sites up in northern Michigan, eastern UP. We also started a pilot project at Oshkosh, which I'll have one slide on, and then also down in Cleveland. And these projects are focused on looking at the responses of the ecosystem to these different management techniques, and also we're looking at nutrients and how harvesting affects nutrient cycling in Great Lakes wetlands. And then the third component is expanding on biomass utilization techniques. So how can we take this biomass, this waste product, turn it into energy or turn it into some sort of compost that can be fed back into agricultural systems, potentially offsetting nutrient inputs into agricultural systems, which is the problem in the first place. So if you can take phosphorus that's in this wetland as a problem, as a pollutant, and then compost it and put it back on farm fields and reduce the amount of external phosphorus that the farmer is pouring onto the fields, then that could close the loop and overall reduce the problems somewhat. So we're looking into that. And here's just a couple images showing the potential for phosphorus removal specifically, or I guess in nitrogen. So based on just simple biomass back in the envelope measures, you can remove approximately 30 kilograms of phosphorus per hectare by harvesting invasive cattails, and 120 kilograms of nitrogen per hectare. This it represents, if we were to harvest 100% Great Lakes wide, how much nutrient we could remove from the Great Lakes ecosystem. So I'm going fast. I realize I've got a lot of slides here. Okay, so zooming in on Cheboygan Marsh, where we have really ramped up our experimental work to a relatively large scale. We put in 60 by 60 meter plots out at Cheboygan, and we're looking at a whole range of different management techniques. So we're looking at an annual surface harvest. We have controls. We're doing a crush treatment because a lot of land managers use crushed cattails as opposed to actually harvesting them for habitat value. We have an annual, repeated annual harvesting, and then a multi-harvest where we come in every couple times a year and remove the biomass. And in 2015, we harvested 30 acres of cattail experimentally at Cheboygan. We're looking at sediment, pore water, nutrient chemistry, plant community responses, and greenhouse gas emissions from all these different treatments. And then we're also utilizing biomass. Approximately 50 tons of the biomass is being composted by Carl Remans, who's a local guy close to the biostation. And 5 tons of that biomass is being converted into biofuel pellets. And we're hoping to ramp up the biofuel pellet development next year. A second study that I'm actually not going to go into, beyond just saying that we have a variety of other harvesting-related studies occurring at those different sites throughout the Eastern UP. Okay, so another aspect of our work that I think is really exciting, I think there's other people at the biostation that are interested in this type of work, is looking at drone imagery and trying to use high-resolution imagery to develop surface models and trying to get at some of the structural heterogeneity of the marsh ecosystems. So I think there's potential. We've got money to hire someone to fly for a week some of our sites this coming summer, so there might be some potential to connect with other people that are interested in getting some of their areas flown, because we've got a pilot and drone coming in, so there might be some potential there. But this is just a couple of images from Cheboygan, and thanks to Jason Talent for actually creating the digital elevation models for us. This is showing us, this is a digital elevation model, and you can see our different plots after they were implemented, and these in green are our control plots. You can see they have very clear elevation was picked up between the harvested and unharvested. So that gives me some confidence that this is actually a pretty accurate model. Okay, biomass utilization. This image just shows kind of the whole process from harvesting biomass in the field, creating big piles of it, and then we have a grinding apparatus that pounds it into little pieces, and then we have a pelletizer where we can condense that biomass and turn it into these pellets that can be theoretically used in a home heating system or in like a boiler or something to create energy. Okay, so in addition to the work at the biostation, like as I said, we have these other sites. Really exciting working with Bob Pillsbury at University of Wisconsin Oshkosh, where they have a lot of invasive cattails. We did a small couple-acre harvest this year. And the other really great thing about Oshkosh is they had an anaerobic digester on site that provides something like 10% of the electricity for the campus. And so we spent a day, I think, harvesting out there this year, and took all that harvested biomass and plugged it into their anaerobic digester and turned it into electricity and heat that is then being used on campus. This is just the tip of the iceberg on this. I mean, there's a lot more potential at Oshkosh. Similarly, we did a pilot project down in Cleveland with the Cleveland Museum of Natural History, where we harvested about 3 acres of invasive Phragmites, and you can see how tall that stuff is. Our harvester is about 12 feet tall, so this stuff is like, you know, at least 12 feet tall. And the biomass that was harvested in Cleveland was, this is all the whole process, taken to another industrial scale anaerobic digester facility, and biogas and electricity was created. Additionally, they also have a liquid fuel pumping station at this site with natural gas, or effectively natural gas. Okay, a lot here. So I'm really excited about this too. We have, just in the last month, we found out we're getting 2 additional grants that are going to ramp up our work for the next 3 years. And yesterday I was told that I should not say who the funder is for this one yet, so that's what this little blue box is. But I was not going to take it out of my presentation. So this is a partnership with Oregon State University, with the Sioux Tribe Chippewa Indians, Beth Lawrence at the University of Connecticut, Dartmouth College, Nick Rio at Dartmouth College, who was a former UMBS student, and he has Native American Studies, environmental science, and then Amy Schrank at Michigan Tech. And this is an exciting project because we're going to be looking at, sort of reminds me of the dam removal project. We're looking at many different aspects of biodiversity response to harvesting. So we have essentially, we're going to be looking at including some fish passageways into our treatments, and we're going to be monitoring plant communities, fish, macroinvertebrates, herps, birds, greenhouse gases, and then we're also going to be doing drone flights of all these areas to measure some structural heterogeneity. Almost done. Here's just an image showing kind of what we're looking at with these aquatic connectivity plots. Passageway into some of these cattail stands. Okay, and then the second last thing I'm going to talk about is the second project we were just funded from the Michigan DNR, and this is a 2-year project partnering again with many of the same people, and also Boise State University remote sensor Jeremy Grant, who's there, and we are going to be looking at novel approaches to another invasive plant, European frogbit, which has shown up recently in the same areas we're in. So this project has 2 components. It has a remote sensing component, and it also has a management component. So it's kind of similar in structure, but that's a couple-year project. Okay, so again, it's just collaboration potential. There's a lot of overlap. There's all of this expertise in this room with different taxa, and I think there's potential with classes to plug some of that work into our already experimental stuff that's going on. And we also have these regional partnerships. We've really connected with a lot of different groups. So for other people who are looking outward, we've just kind of really plugged into a lot of the local work. Okay.
Thank you, Shane.